Imagine you're trying to build, I don't know, the world's most sophisticated supercomputer. You've got all the best parts, the best processors.
The most advanced memory chips, everything.
Everything. You assemble it perfectly, piece by piece. But then you realize you've forgotten one tiny crucial detail, the power cord.
And suddenly you have a very expensive, very beautiful paperweight.
Exactly. It just sits there. It looks impressive, but, eventually the battery drains and it all just shuts down.
And that is essentially the exact problem scientists have been banging their heads against for years when it comes to growing human eyes in a lab.
Or, to be more specific, retinal organoids.
Right. These are these tiny 3D balls of tissue grown from stem cells that are meant to mimic the human retina.
And we can grow the cells. We can make them look like a retina, but we've never been able to give them that power cord or, I guess, a plumbing system.
Without blood vessels, the most important cells they starve and die.
That's been the bottleneck for a decade.
It really has, until now.
Until now. Because today we are diving deep into a truly groundbreaking new study from Cell Stem Cell. It was published just this week, February 2026.
The paper is by Sharma et al. And, what they've done, it really sounds like science fiction.
They bioengineered a human retina that grows its own blood vessels. Then they connected it to a microfluidic gym for its nerves.
And kept it alive long enough to actually process complex visual signals.
And we are not just talking about cells twitching in a dish here.
No, not at all. We are talking about the first time we've seen a lab-grown retina develop on, off, and even on-off signaling pathways. That's the complex circuitry you need for real vision.
We have a massive stack of data to get through, so our mission today is to deconstruct this paper, sentence by concept. We're going to look at the engineering, the biology.
And, of course, the incredible implications this has for curing blindness.
Okay, so to do that, we need to introduce the two main characters in this cellular drama. First up, the victim.
The retinal ganglion cells, or RGCs.
I like to call them the VIPs of the eye.
They certainly are. RGCs are the neurons that take all that visual information from the eye and then send it down the optic nerve to the brain.
So if your RGCs die, you go blind. It's really that simple.
And in the standard lab models, they are always the first to die.
And then we have our hero.
The hero of the story. Induced endothelial cells.
Okay, so these are the plumbers.
Exactly. Endothelial cells are the cells that line our blood vessels. And in this paper, the researchers didn't just hope they'd show up. They forced them into existence.
So let's unpack that problem first. Why has this been so difficult? Why can't you just grow a retina and have it work? These organoids are floating in a nutrient-rich liquid, right?
They are.
So why doesn't the oxygen just sort of soak in, like sponge?
It all comes down to physics, specifically the limits of diffusion. A standard retinal organoid, or RO, is a sphere. It's a 3D ball of cells.
Okay.
And as it grows, the cells on the outside have no problem getting oxygen from the liquid. But oxygen can only passively diffuse about maybe 200 to 400 microns into tissue.
So once the ball gets bigger than that?
The cells in the very center are just too far away from the surface. They start to suffocate.
And that state is called hypoxia.
Yes, and hypoxia triggers a self-destruct sequence.
A process called apoptosis.
Apoptosis, or programmed cell suicide. And unfortunately, just because of the way the retina develops, those retinal ganglion cells are VIPs. They're located right in the core of the organoid.
So just when the retina is starting to get interesting, the connection to the brain dies off because it can't breathe.
Precisely. Sharma and his team realized that to save the RGCs, they had to bring the oxygen to the center. They needed to build a vascular network.
But you can't just, inject tiny blood vessels into a microscopic ball of jelly.
No, you can't. You have to reprogram the biology itself. So the researchers started with human-induced pluripotent stem cells, HIPSCs.
These are the stem cells that can become anything.
The blank slate cells. And they used a specific genetic switch called ETV 2.2 isoform 2.
Okay, let's pause on that term. ETV 2.2 isoform 2. That sounds like a software update.
In a way, it is. It's a transcription factor.
Right.
So think of your DNA as this huge library of blueprints.
Right.
A transcription factor is like the librarian that goes and pulls a specific blueprint off the shelves and says, build this.
I see.
So by activating ETV 2.2, they forced these blank slate stem cells to become endothelial cells, the vessel builders.
And just to make sure they could see what was happening, they tagged these cells with EGFP.
Enhanced green fluorescent protein. Essentially, they made their plumbing crew glow neon green so they could track the whole construction process under a microscope.
So they have the retina parts and they have the glowing green infrastructure team. Now they have to put them together. And I assume you just mix them in a beaker and hope for the best.
You'd think so. And that's exactly what they tried first. They called it simultaneous co-culture.
So all the ingredients in the pot at day 0.
Yep. And it was total chaos. It turns out retinal cells are incredibly picky. They need to form very specific, delicate layers to function. And when you introduce these aggressive vessel-building cells too early, they just barge in and disrupt the whole architecture. The retina couldn't organize itself. A complete structural failure.
Okay, so timing matters. You can't put the plumbing in before you've even poured the foundation.
Exactly. So they tried waiting. That was attempt number 2. They grew the retina for a little while, and then they added the vessel cells mixed inside.
Did that solve the chaos problem?
It solved the chaos, but it created a yield problem. The vessel cells just didn't really take hold. They didn't integrate well enough to save the tissue, so they were striking out.
And that brings us to combination 3, the winner, the carpet method.
This was the big breakthrough. So they used a standard protocol to start growing the retinal organoids. Then, at day 7, instead of mixing the cells inside, they placed the tiny organoid balls on top of a flat layer, a monolayer of these endothelial cells.
It's like planting a bush on a nutrient-rich mat.
That's a great analogy. They let the organoids sit on this carpet of endothelial cells. And remarkably, the endothelial cells started to migrate up and into the organoid.
So they crawled up into the tissue on their own.
They infiltrated it naturally, like roots growing into soil, but in reverse.
That is such a cool visual, these green glowing cells just weaving their way up into this little ball of retina.
And because they entered naturally and at just the right time, they didn't destroy the delicate layers. They wove themselves in between the other cells.
Okay, let me just pause you there because this carpet method really sounds like the pivot point of the whole study.
It was.
I want to make sure I'm picturing this right. So we started with the deadly center, the fact that these mini retinas suffocate from the inside out.
Right. Hypoxia kills the ganglion cells.
Then Sharma and his team genetically hack stem cells using that ETV 2.2 thing to become blood vessels.
Right.
But instead of just mixing them in, which causes chaos, they lay them out flat like a placemat. And the retinal ball sits on top and the vessels grow up into it.
You've got it. That's the perfect summary. And once that plumbing was installed, well, that's when things got really exciting.
So visualize those green fluorescent tubes winding their way into the center of the sphere. The plumbing is installed, but does it actually work?
That is the million-dollar question.
Right. It's one thing to have cells that look like blood vessels. It's another thing entirely to have functional pipes. If I glue a garden hose to a wall, it looks like plumbing, but water isn't flowing through it.
Exactly. So once they saw these green networks formed, which happened in about 72% of the organoids by week 3, they had to test them. First, they looked for a marker called PCAM1.
PCAM1.
It's a protein found on the surface of endothelial cells. And it just confirms, yes, these are in fact vessel cells. But to test if they were hollow tubes.
If they were luminized, is that the term?
Luminized, yes.
Yeah.
To test that, they do what they call the open window test.
I love this experiment. They used a dye, right? A red dye.
They did. Dextrin 598. It's a fluorescent red dye. They put it in the liquid outside the organoid. Now, if the vessels were just solid cords of cells, the dye would stay on the outside.
But if they were open tubes connected to the surface,
the dye should flow inside. And it did.
No way.
The red dye ended up deep inside the green loops in the center of the organoid. The vessels were hollow, there were open windows, allowing nutrients to passively diffuse deep into the tissue.
That's huge. So even without a heart pumping blood, just having these open tunnels lets the oxygen get to the center.
And they proved that with another visual test. They used a hypoxia reagent, a chemical that glows green only when oxygen is low.
So green means I can't breathe.
You got it. In the control organoids, the ones without vessels, the centers were glowing bright green. They were suffocating.
And in their new ones, the VROs,
the glow was significantly dimmer. The plumbing was working, oxygen was getting in.
And because the oxygen got in, the VIP survived.
The difference was night and day. They tracked the retinal ganglion cells using a different transcription factor, this one called POU4F1.
Okay, so POU4F1 is the ID badge for the ganglion cells.
That's right. And by week 18, that's 4.5 months of growth, the standard organoids had almost no RGCs left. The count was 1.2%.
1.2% is basically extinction.
It is. But in the vascularized organoids, the VROs, the RGC count, was 18.2%. It was a massive, massive preservation of that critical tissue.
Okay, so we've engineered the vessels, we've saved the cells, but a neuron that's alive but totally disconnected is, well, that's just a lonely cell. It needs to connect. It needs to do something.
And that brings us to the next big hurdle they had to overcome. In a ball of cells floating in liquid, the RGCs have this nowhere to go problem.
Right. Their output cables, the axons, they would normally grow towards the brain. In a dish, they just tangle up and die off.
The cells are alive, but a neuron with nowhere to send a signal is just, well, it's screaming into the void. How do you solve that connection problem in a plastic dish?
You have to trick them. You have to give them a destination. The team used a microfluidic device, which you can think of it like a two-room apartment for cells.
Okay, I'm picturing it.
In the living room, you have the retinal organoid, but the wall separating it from the bedroom has these tiny microscopic tunnels carved into it.
Like little mail slots or something.
Exactly. And because the neurons are just desperate to connect, they send their axons, their output cables, threading right through those tunnels into the next chamber. It aligns them. It creates a physical optic nerve out of living tissue.
And that device also allowed the researchers to eavesdrop on the cells. They used what are called microelectrode arrays, or MEAs, in the floor of that second chamber.
And what they heard was the chatter of a living network. The VROs weren't just sitting there, they were firing action potentials. These are the electrical spikes.
Faster and stronger than the controls, I assume.
Much faster. But more importantly, they saw bursting and synchrony.
Okay, explain why synchrony is the key here.
Well, if individual cells are just firing randomly, it's just noise. It could be irritation. But if they fire together in coordinated bursts, it means they are talking to each other. It suggests a functional network.
And they proved they were physically connected by, what, trying to hang up the phone?
They did. They used a drug called carbonoxylone. This chemical blocks what are called gap junctions, the physical bridges between neurons. When they added it, the synchronized bursting just stopped.
So just to lock this in, the dye test proved the vessels were hollow, solving the oxygen problem. The microfluidic chips solved the tangling problem by guiding the axons.
Right.
And the electrical readings proved the network is actually talking to itself.
Exactly. We have a living, breathing, humming retina, and this is the big one. The ultimate test of an eye isn't just, is it alive? It's, can it see?
And this is where the paper gets incredibly sophisticated.
Right.
They had to distinguish between, let's call it cheating and real vision. Well, biological cheating. It's a technique called optogenetics. To make sure the RGCs were even capable of firing in the first place, they genetically modified them with a protein called F-crimson.
F-crimson, it sounds like a spell from Harry Potter.
It basically acts like a cheat code. It makes the RGCs fire whenever they hit with yellow light. That's 580 nanometers. It completely bypasses the rest of the eye. It doesn't matter if the photoreceptors work or not. You hit the RGC with yellow light, it fires.
So that proves the output cable works. But what about the input? What about the camera itself?
For that, they had to wait.
Yeah.
They kept these organoids alive all the way until week 31.
Wow, that is over 7 months.
It's a huge investment of time. But by week 31, the actual photoreceptors, the S cones and M cones, should be mature. So they ran the critical experiment. They flashed blue light, 470 nanometers.
Why blue light specifically?
Because the cheat code, F-crimson, doesn't respond to blue light at all. But the S-cone photoreceptors do. So if the RGCs fire when you flash blue light, it can only mean one thing.
That the signal traveled the long way around. Light hits the photoreceptor tells the bipolar cell tells the RGC.
It proves the entire vertical circuit is connected and functional.
Wow.
And not only did they see a response, they saw the holy grail of retinal processing, on-N, OFF, and on-off-F responses.
Okay, I really want to double-click on this because off-off responses are a huge deal in neuroscience, right?
They are. A simple light detector just says light is here. That's an on-N response. That's easy. But our eyes are so much smarter than that. Some of our cells fire when the light turns off.
Wait, so seeing darkness is actually harder than seeing light?
Biologically, yes. Think about the logic behind it. To fire when the light goes away, you need an inhibitory neuron in the middle of the circuit. You need a NOT gate. So light hits the receptor tells a middleman cell, shh, be quiet. And then when the light stops, the middleman is allowed to yell.
That requires a much more complex chain of command.
Exactly. It requires specific inhibitory synaptic wiring. It's incredibly hard to grow in a dish because it implies such a high level of developmental maturity.
And they saw this?
They saw it all. They saw on-end cells that fired when the light flashed. They saw O-off cells that fired when the light stopped. And they even saw O and O-off cells that fired at both changes.
That means the organoid isn't just detecting photons, it is processing information. It's detecting contrast.
Correct. It demonstrates that these vascularized organoids had reached a level of maturity and circuit complexity that we just don't see in standard models. It's a huge leap.
That's absolutely wild. We essentially have a biological camera processor growing on a chip.
A human one. That's the key.
Yeah, there's one last segment of this paper that I found fascinating. After building this beautiful, perfect model, they decided to break it.
Well, science is often about breaking things to see how they work, or in this case, how they fail. They wanted to model a specific disease, retinopathy of prematurity, or ROP.
And this is the condition that can blind premature babies?
Yes. In premature infants, the blood vessels in the eye aren't fully formed. When the baby's put on oxygen, the vessel growth just stops. Then when they come off the oxygen, the eye panics and starts growing vessels way too fast. It's called neovascularization.
So you're basically torturing these little organoids to see if they break in the same way a human eye breaks.
In a controlled way, yes. They took their healthy, vascularized organoids, and they put them in a low-oxygen chamber, only 4% oxygen, and they added a growth factor called VEGF.
Recreating the panic mode.
And the organoids responded exactly like a human eye with ROP. The vessels went haywire. They saw this rapid, disorganized overgrowth. They saw increased expression of PECAM1 and another marker, VWF.
And they saw something called empty sleeves.
This is a very specific, very telling pathological sign. They saw the structures, the scaffolding of a vessel made of something called COL4A1, but there were no endothelial cells inside.
Ghost vessels.
Exactly. The vessels had grown and then regressed or died, leaving this empty sleeve behind. This happens in human pathology. Seeing it in the dish confirms that this is an incredibly powerful model to test drugs for ROP.
So instead of testing on animals, or heaven forbid babies, pharma companies can test therapies on these ghost vessels in the lab.
That is the power of this technology.
So we have covered a lot of ground. Let's try to bring it all together in a final synthesis.
Sharma and his team, they really achieved a trifecta here. First, the bioengineering. They successfully integrated functional hollow blood vessels into human tissue using that carpet method.
Right. Then there was stabilization.
Using the microfluidics to save the axons. And finally, functionality. They achieved long-term survival over 31 weeks and complex light processing, including those on and off F pathways.
They basically built a human retina on a chip that actually works. But I have to ask, is there a catch?
There is a limitation, and the authors are very upfront about it. These vessels are transient.
They don't last forever.
They eventually regress. I mean, remember, even though the vessels are hollow, there is no heart. There is no active blood flow pumping pressure through them. And without that mechanical stress, that constant flow, blood vessels just tend to break down over time.
So the plumbing eventually gets clogged or collapses because the water isn't running.
That's a great way to put it. Future work needs to connect these organoids to some kind of micropump system to simulate a heartbeat. That might keep the vessels stable for years instead of months.
Still, even getting to week 31 with this level of fidelity is just, it's a massive leap forward.
It completely changes the baseline for what is possible in vision research.
And so before we sign off, I want to leave our listeners with one final thought. We've talked about on and off pathways. We've talked about these organoids processing contrast and edges.
Okay, where are you going with this?
Well, if we solve that flow problem, if we can keep these vessels stable for years, and if we connect those microfluidic channels not to a computer, but to a brain organoid, you're asking when the model stops being a model and starts to actually see.
Exactly. If it has the hardware and it has the processing power, at what point does it stop being a clump of cells and start being an eye?
That is a question that really blurs the line between biology and philosophy. But, technologically, we are getting closer to finding out.
And on that slightly terrifying but awesome note, we want to hear from you. If you could use this technology to cure one specific type of blindness, but it required growing your own spare biological parts on a chip for 30 weeks, would you trust the ghost vessels?
Send us your thoughts. We'd love to hear them.
That's it for this deep dive. Thanks for listening.